Today I'm going to tell you the story of my life and the structure and property of water. Now here I am back in 1966, and we had done some studies about water and magnets, and we found that water is repelled by a magnetic field. Whether it's north or south, we found that water has a slight diamagnetic property, and it's repelled by a magnet. Water is a polar substance, so it has a north and a south pole. And then I started doing work, and I published a study in '66, the magnetic effects on evaporation. Now, this study has been reproduced later, but I was, as far as I can tell, the first one that really had done a study of this. And now we started to see that it's exciting. See, water is not alive, but water is reactive, and it acts as if it is alive. Here's a little bits of food coloring. You see them playing with each other, dancing around, and no other energy, just water, ionic attraction and repulsion. And then we found out, you see, blood is also repelled by a magnetic field, north or south, because blood is so much water, or for whatever reason, it's not attracted, it's repelled by a magnetic field. Very interesting. Out of high school, I was hired as an electrical engineering student. I worked on the Apollo project. In fact, I was very helpful in bringing them back, the Apollo 13 astronauts. This was the time that I actually got exposed to beryllium, got beryllium in my lungs. And I had a host of other scientists that I'd worked with and started to broaden my ideas. But let's get back to water. You see, water is a very, very special substance. Has many different types of things that make it unique in the chemical world. And we have to try to understand it more. And our bodies are 75% water by percentage, but by actual molecules, 99% of our bodies is water. So I started to find out that water is a liquid crystal. And I started to understand and research liquid crystals. Water, between the temperatures of 0 and 60 degrees, has a liquid crystal. And that crystal can absorb a memory. Now, this is also due to that magnetic polar effect of water. So, recognizing that if this is an electric crystal, so what if I froze the water? Well, I got this type of a microscope, the Chiral liquid microscope, that would be able to freeze the water as I was able to look at the crystals starting to form. And the first form of this study was done in the 80s. We published it in 1984. We're able to see that we can now find the structure of a homeopathic, because the homeopathic is affected by its liquid crystal structure of water. So as we started to formulate and get these crystals started in water, I found I could measure what was a homeopathic, that I could tell which homeopathic it was by looking at the crystal structure. In 1986, I lectured in Japan. Masaru Emoto came to that lecture, and we discussed how to make other different factors. He started doing work, and he found that he could put emotions into the water. I found that it was really difficult because of the individual tendencies. However, if we started to get more people, we could get better quality. Masaru Emoto's work became world famous. People seem to forget about me and how I started it. But I tried to really help him in many different ways. And I told him that we have to broaden it so that we can get better ideas.
Water memory. Yes, you heard it right. Water can remember things, and even recreate certain properties of what it remembers. In the 1960s, in a closed lab, something inexplicable occurred. A lab assistant dropped a vacuum-sealed ampule of poison into water, and the tube was removed from water only after 3 days. This water was given to the lab mice to drink, which resulted in the death of all the mice that drank the water. Immediately the water was chemically tested, and it was still only water, with no trace of poison in it.
Water has memory. Well, extraordinary claims need extraordinary evidence. I've always believed in this and tried to publish and do the science and publish that and get some critique. It's not always easy. So we're trying to find ways to control, quality control homeopathics. And another technique we tried was the Curlian photography. Here we used a ball, plasma ball, with rare electron gas. This would make a rare electron gas. I called it an allopathic evaluation, or Reg-A. And now I could take pictures of homeopathics, and yes, lo and behold, we could find that we could measure differences, subtle differences, inside these different pictures. Some of them were dramatically different, but we could start to see that there was a way to look at the energy state. When we did this with different people, we found out that there was a variance between people. So that created a kind of a problem, getting quality readings. Then Benveniste had written me, said he didn't quite understand electrochemistry or quantum theory. I tried to help him to explain exactly what electrochemistry was. He set off on doing some experiments regarding an antihistamine compound and how it could be transmitted into water, and at high dilutions where there was no original substance, that he would find results. And he started doing these experiments, and he got results.
So they carried out hundreds of experiments and soon realized that they'd made an extraordinary discovery.
The structure of water means how its molecules are organized. We can see how water molecules join together into groups. These are called clusters. Scientists came up with the idea that these clusters work as memory cells of a certain sort.
It seemed that when a chemical was diluted to homeopathic levels, the result was a special kind of water.
Water, as a liquid crystal, would be able to generate some crystal shapes through the succussion process to help remember the shape of a substance.
It didn't behave like ordinary water. It acted like it still contained the original substance.
The ancient mystics have told us for ages that water has memory and intelligence. Water is the key of life, but it's also the key of homeopathy.
It was as if the water was remembering the chemical it had once contained. So Benveniste called the phenomenon the memory of water.
Water has a polarity to it, a type of magnetic action, which allows for a liquid crystal state. Information or shapes can be transferred into this liquid crystal, and this is what homeopathy ultra-high dilution is.
Water recalls the whole history of its relationship with the world as if on magnetic tape.
At last, here was scientific evidence that homeopathy could work.
Now, if you watch the movie about the misunderstanding of homeopathy, you see how Benveniste was betrayed by Randy, a lying, cheating magician, and there was something there. Something there. Well, then my life led me to Kiev. Let me tell you the story of our cancer studies in Kiev, proving natural medicine.
Breast cancer, horrendous disease. Here we have a young girl affected. Chemotherapy was discovered from natural medicines. Natural medicines that fought cancer. Chemotherapy was then derived from these natural medicines. But yet no study had ever been done on natural versus the synthetic chemotherapy. No study, that is, until we did our study in Kiev.
My first visit to Eastern Europe was at Kiev in 1991. Now, this was just after the 1989 dropping of the wall, and the societies were in crux. I was invited to the Institute of Oncology to give a lecture about energetic medicine. And what I found is that the society just didn't even have money. They used coupons. So I found that I could hire some of the best doctors in the world very, very cheaply. $10 a month, seriously. And I hired some of the best doctors at the Institute of Oncology. Doctors who had fantastic minds working in hospitals, recognized by the World Health Organization. Fantastic, incredible opportunity for us to do natural medicine testing to see if we could find out if natural medicine, because drug companies won't do this study. Now, basically, the discovery of chemotherapy comes from vinca. They found that this is a poisonous plant that animals will not eat. Animals will not eat this plant because it's poisonous. They have an instinct. But when an animal, one of the farm animals, gets cancer, when they get cancer, all of a sudden then the farm animal wants to have the vinca. The farm animal will eat it. The farm animal, the horse, the cow, they'll break down the fence to get to the vinca when they have cancer. Only when they have cancer. They don't do it when they're healthy. You see, because there's a sense inside. And all of a sudden the cancers go away. So now the drug company started to see that this plant could kill cancer. This plant has a poison, but the poison is much stronger against the cancer cells. So this is the basic development of the simple ideas of chemotherapy. Now, nobody had ever done a test of whether the natural chemotherapy and how did it work versus the synthetic, because the synthetic was designed for the patents. But here was my fantastic opportunity at the Kiev Institute of Oncology to test this in a hospital. First we did cell culture tests. Then we moved to animal population tests. To be able to do this properly, we had to follow the protocols. And I had to fund the entire operation, basically myself. And then lo and behold, here we found the actual test. We went to the patients and did a patient trial. There was 120 patients in each group, and basically we were able to study the effects of synthetic chemotherapy versus the natural chemotherapy. The real vinca versus the synthetic vinca. And lo and behold, what we were able to find is that the real vinca is incredibly safe and incredibly effective.
Not only was the natural medicine much more safe, but it was dramatically more effective.
Yes, history was made. Natural medicine was more successful than the synthetic. And this was published in a peer-reviewed medical journal. So then we started to look at other different studies and started to look at an expansive viewpoint. And we did other studies on the medicine and found out that there was incredible results. Kiev was pretty rough, so I moved to Budapest and decided to work with natural medicine. I was hired as a medical doctor, professor of medicine at Semmelweis University, the largest, most established medical university in Budapest. And I was there to try to help people. Now here's scenes from the true story, Water, Wine, Homeopathy, which you can watch and learn more about the intimate story. My son Sterling was born, and actually he was kidnapped. We just got him back on this day. You're actually looking at the time we got him back. And this is my ex-wife, Ildiko, with Sterling. And we were able to do incredible research. And then I documented this in film and in books. And here is the very, very first time I touched and met my daughter, Destiny. Now, our research was approved for a presentation at the University of Exeter in the Complementary Health Symposium. And then at the very, very first European Association for Clinical Pharmacology, the theme of the Congress was chemotherapy. We get you better, but you come back. Why? At the beginning of my address, I said to the Congress, I think I have the answer for you. I think I know why. You see, our results were clear. Chemotherapy gets you better, but the chemotherapy weakens your immune system, so you come back. You do not have cancer because you're chemotherapy deficient. You get cancer because you're immune deficient. The natural treatment group increased the immune systems. They had less cancer in the future. Chemotherapy gets investment for research because of patent protection for profit. The natural medicines work better, but the drug companies want to protect their profit, protect their patents. So the investment of the research is done on the chemotherapy, the synthetic drugs.
I heard a presentation at the pharmaceutical college. You're really against the drug companies. But the drug companies buy pencil, paper, and computers. What will you do without them?
Well, I think we have to think about the patients. I think we got to start thinking about the people. The drug companies are causing too many side effects. I believe that natural medicines can take their place. I know there's a lot of money involved, but I believe that what the people want is more care about them rather than care about their paper and pencils.
Well, you may be right, but you are going to cause a lot of trouble for yourself and for a lot of people.
Well, sorry about that. You want some peanuts?
No, thank you.
I made the movie Water, Wine, Homeopathy, where every scene opens with water, wine, or homeopathy. And I wanted to tell the true story of what happened, because once I had stepped on the toes of the drug companies and their profits by proving that natural medicine was not only safer but more effective, especially in chemotherapy, I became very much a problem. And I was beaten up, and they tried to kill me. Luckily, I was able to escape, to be able to tell the story. Now, the persecution, the prosecution, and the character assassination that they've done on me is incredible. And I want you to realize that really natural medicine works, and there is a shape of water. So I am Desiré Dubonnet, the man who left America to find freedom, who's no longer a man. And when I became Desiré Dubonnet, the drug companies stopped trying to kill me. They figured you won't take me serious. You can learn more about our research in voltammetry, but back to magnetic, we found that certain bacteria have magnetic properties, and by changing magnetic fields, we could change their motion. So we started to understand more and more, and we set up this experiment to be able to magnetically affect the bacteria. Now, the great quantum physicist, Erwin Schrödinger, told us there's a negative entropy, that there is an energy factor in life. And once we got these bacteria, then we could get them to line dance.
Where did you come from? Where did you go? Where did you come from, Cotton Eye Joe? Said I've been to Cotton Eye Joe. I've been married long time ago. Where did you come from? Where did you go? Where did you come from, Cotton Eye Joe? Said I've been to Cotton Eye Joe. I've been married long time ago. Where did you come from? Where did you go? Where did you come from, Cotton Eye Joe? Said I've been to Cotton Eye Joe.
Our research in voltammetry, electrophysiological reactivity, and these different things, we were able to publish in 1997. And we have done all this research that took years and years to compile and to do. I started with research that I did on AIDS patients at a Hungarian hospital. I was able to test all of the AIDS patients in Hungary, HIV positive. And we found that there was a reactive pattern. To publish this, I was invited to Singapore, to the International Congress on Sexually Transmitted Diseases. There, I would be meeting the famous Nobel Prize winner who discovered the AIDS virus. So in 1997, I went to the World Convention and was able to meet Luc Montagnier. He heard my lecture on the electrophysiology. He heard my lecture on the energetic medicine. He heard what I was doing, and basically he said this was the future of medicine, that we needed to get away from the synthetics, and we needed to start looking at the energetic factors. So him and I communicated about the work that we were doing on measuring the electrophysiology of homeopathy and the electrophysiology of our patients. And then he went on to do work into water. Let's look at some of the work he did.
Water has the ability to reproduce the properties of any substance it once contained. Water would have the ability to retain a memory of the molecule's properties.
Water, we said, is a polar substance. Thereby it has north-south poles and can organize itself in some type of a structure. This liquid crystal effect would be able to generate memories, but it would be kind of unreliable. Luke took a sample of HIV-infected DNA, and he was able to then put it into a test tube, and then diluted, homeopathically, as I explained what to do. 1 part to 10, 1 part to 10, till he didn't have any of the original substance left. But then he used a type of microphone induction magnetic coil to absorb the information and send it to the place in Italy. They were measuring the background noise, and you can see that there's a little bit more background noise today, because they had some cameras that were shooting the scene. Now, water can take a memory through its liquid crystal effect, but there are still lots of variables that are happening in this liquid crystal. Not a true liquid, but definitely not a true crystal. So when they were measuring this, as Montagnier and his team were measuring it, they used this little type of induction coil microphone. Not really a very precise instrument. And they got results, though. They could see results. But I'm telling you that when you get to water and you're trying to analyze water, measuring the magnetic flux electrically isn't quite the best way.
Starting with the DNA of an HIV-infected patient, he will create a digital file, send it through the internet to another lab where the DNA will be reconstituted from that digital file. The professor calls it transduction. We could almost call it teleportation.
Montagnier and his team were not electrical engineers. They were using this type of induction coil to measure a flux, but really that type of flux takes more of a motion, and it's not really accurate. They didn't have the scientific engineering technical expertise. Let's follow the story. Freezing, curling photography, electromagnetic coils weren't enough. We needed to do conductivity.
All sharks are attracted to the weak electromagnetic fields of their prey, but it's thought that the hammerhead's extra-wide head shape gives it an especially sensitive detector. To find out just how sensitive, Steve is using electric currents to create these fields and see what field strength they can detect. The circle of cable on the left has a current running through it, while the one on the right has none at all.
How minor is this current we're talking about? This current that you're putting in here?
Right.
How minor is it?
What I'm applying right now is 7.7 microamps, 5 nanovolts per centimeter, 5 billionths of a volt per centimeter.
It's an incredibly small figure.
The way to think about that would be to take a standard flashlight battery and stick one lead in the water in Boston, and the other lead in the water in Miami. And a shark swimming directly between those two leads would be able to detect that minute amount of current flowing between those two leads.
Just a little torch battery.
That's right, that's right.
And that's about 1,500 kilometers or so.
Right, what's that? Yeah.
Scientists have said that there's an electrosense in humans in the nasal pharynx, in the taste receptors and the smell receptors. So I recognized we needed to use electrodes, voltammetric electrodes, and I had to study voltammetry to really understand how the nose acts as a voltammetric field detector to find things, like the shark does in the contact of water, but we would have to contact through smell. Having done this, I got it registered with the FDA 1989. Amazing. Using electrodes to put into the substances and electrodes to measure the human body, we could measure the voltammetric field of a substance. Using this device here is what we use now, after a long evolution. This is basically known as an electric nose and electronic tongue that would give us an ability to measure the voltammetric electrical fields. Here's a sample that we're measuring a substance. And as we run in a voltage, we're going to detect amperage changes, as vice versa. And being able to find out a true electrical signature, an electrochemical signature of the shape of the item that we are testing. This would allow us to take this in. We'd be able to study this. We started studying this way back in the 80s. All different types of studies validating and verifying the effects of the voltammetric field and able to do this. This is dramatic, evidence-based, published in peer-reviewed medical journals, and yet still rejected by the chemical companies who don't like work that challenges them. In the year 2000, I lectured at the Nobel Prize Hospital in Stockholm, Sweden, about this. Now, we've seen that there's structure in the memory of water. Many other people have written scientific articles about this. Other people have expressed and redone the studies of Benveniste, Montagnier. But many of this is really hard to reproduce and to get quality work. We published our work in the electrophysiological feedback measures of the interstitial fluids, and also here in the structure of water. I welcome you to get these books and read more. You see, water is so important, and one day in the summer when I forgot to water a plant, here we have what happened when we gave water. You see, the water is a beautiful thing. Water can help restore life. Water can help to bring things back to life. So it's amazing. We need to learn more about water, and the shape of water, and the memory of water. And this is Desiré Duvenet. I hope you've learned a little bit more about water and some of the things that I've done to help bring water and the shape of water to science. Everyone who's ever tried to prove water memory is attacked violently by the drug cartel, the evil ultra-rich. But I'm not the kind to give up easily.